Hello, class. In this lecture, we're going to talk about the history of microbiology, some of the key discoveries, and individuals involved in those discoveries. By the end of this lecture, you should be able to explain the distinction between a pathogen and an opportunistic pathogen, compare and summarize the theories of biogenesis and spontaneous generation, describe the germ theory of disease, use Koch's postulates of disease, and identify the goals of aseptic technique and explain why it's important in healthcare facilities and laboratories. All right, so here is a timeline of like the history of microbiology from around 1665 to 1945. We're going to focus a bit on that golden age of microbiology and some of the key findings in there. But we will start at the beginning. So we would not have microbiology without microscopes. And one of the first descriptions of cells was presented by Robert Hooke, who had an early microscope pictured here on the right-hand side. And that was perfected upon and refined by Antoine van Leeuwenhoek, who was, I believe, a merchant who needed to make microscopes to be able to look at thread quality in garments, but also the first to see bacteria, and thus like the grandfather of microbiology. Now, with these ability to see these organisms that we could never see before, there began to be this debate between the idea of spontaneous generation, which is that life comes from non-living things, or biogenesis, which is that life emerges from existing life. And we had never been able to see this microbial life before, and seeing that made it clear that there were things going on in the world that we weren't able to observe before. And so one of the key ideas or key examples for spontaneous generation is the idea that if you leave out some food, then you're going to get the formation of maggots and flies, right? So there is this Italian scientist, Francesco Redi, who set out to show that this is not because you have some sort of spontaneous generation of maggots on spoiled meat. It's because the flies go in there and lay their eggs, which develop into maggots, which develop into flies. And the way that Francesco Redi did this was he set up this experiment where he had a control, right? So he has this unsealed flask, which is where you would normally see this spontaneous generation, quote unquote, of flies and maggots. He had a sealed flask that was sealed with a cork, and then he had a flask that was covered with gauze. Now, the reason that you have the sealed flask and the flask covered with gauze was this idea that you needed air for spontaneous generation, right? So the cork would block all air transfer. The gauze would allow air to go. Now, what he found is that in both of the covered versions, you get no maggots on the meat, therefore, pretty definitively showing that spontaneous generation is not a thing. But there were some examples that still couldn't really be fully explained, such as spoilage in wine and that sort of thing. And this theory persisted for a couple hundred years after Redi's work. And then you've got Louis Pasteur, who pretty definitively showed that biogenesis is responsible for propagation of life, and that is because he was working with winemakers and trying to figure out how to keep wine from getting spoiled. And he showed that essentially the wine is only spoiled when it's contaminated by microbes. And he was able to show that if you pasteurize your wine, which is basically killing off the microbes, in this case yeasts, that are in your wine, then once they are killed off with this high heat that isn't quite boiling, then you don't get wine spoilage anymore. He had a pretty, nifty experimental setup, which is known as like the swan neck flask experiment. And so if we look in these scenarios here, what he did, which was he used this swan neck flask. So he has this normal flask that has this really long swan neck, right? So it's what this does is it prevents air from really traveling inside of this flask, right? So the dust particles and the microbes in the air get trapped in this bin down here, which doesn't allow them to go all the way here. And so if you take a pure broth and you heat it up, like through this process of pasteurization, and you leave it out, then you don't have any problems with contamination. The broth stays uh, uh, basically sterile. However, if you take this broth, heat it up, and then you tilt the flask to where the broth comes in contact with the microbes and dust that are trapped in that swan neck, then you will get contamination of your broth. Likewise, if you break the swan neck off, you get contamination of your broth. So essentially, what Pasteur was able to show was that if you kill off the microbes in your wine, you do not get contamination of your wine or of your broth. Now, microbes are known to cause disease. They are known as pathogens, but pathogens are a pretty small portion of the overall microbial biomass or microbial species. Less than 1% of all microbes are pathogenic, although there are about 1,400 known pathogens that cause disease in humans. And then there are a handful of pathogens that are known as true pathogens. These are organisms that are obligate pathogens, meaning that that's pretty much the only way they exist. And an example of that would be anthrax. And then you have, alternatively to that, we have opportunistic pathogens, which cause disease only in a weakened host. And an example of this would be like UTIs from a catheter. Now, the germ theory of disease essentially says that microbes can cause infectious diseases. And the pioneer behind, one of the major pioneers behind germ theory is Robert Koch. He developed a technique to determine the specific etiological agent of an infectious disease, or the agent that actually causes that disease. He also did some important early work in the staining and cultivation and isolation of bacteria, and that work is what helped him lead, or what led him to much of his work on germ theory. And so there's something known as Koch's postulates of disease, which has 4 primary steps. All right, and so in step 1, you have to identify the same organism present in every case of the disease, but not present in a healthy individual, right? And so much of his early work was done on anthrax. If you take anthrax, or if you find an animal with anthrax, then every animal that has that disease will have the same organism in it, specifically in this case, Bacillus anthracis. However, that organism is not present in healthy individuals. Now, step 2 is you have to isolate that organism from the host in a pure culture. And then you would re-inoculate a healthy individual with that isolated organism, and that healthy individual would become sick. And then finally, you would re-isolate the disease-causing organism from the newly sick animal, and it would be the same as the previously isolated organism. Now, there are a couple of problems that come up with, while these are very good, like a way of thinking about disease, there are a couple of exceptions and problems that exist here. For example, you have carriers of disease. So in postulate 1, the same organism must be present in every case of the disease, but not present in healthy individuals. Well, you have carriers. These are individuals that are able to have the disease-causing organism in them, but that doesn't happen to cause disease in them. So example here is typhoid Mary. She was a carrier of Salmonella typhi, but never got sick from it. But due to poor hygiene and bad hand washing and that sort of thing, she would shed these bacteria into the food that she made, which would result in the people that she worked for getting typhoid fever and many of them dying. Now, the organism must be isolated from the diseased host and grown as a pure culture. There are a couple of problems here. One is that there are some organisms that are non-culturable. For example, like viruses can't be cultured, and not exactly. I mean, we have some ways of culturing them, sort of. And then you have some organisms that are very difficult or tough to culture. And just because you're unable to culture an organism doesn't mean that it's not the disease-causing agent. And then we have examples of, like, we don't have an animal model, so we can't just reinfect humans with diseases, right? So if there is a organism such as the organism that causes leprosy, that can only infect one host, which is humans, then you can't really test to, you can't use an animal model to test whether or not that works. And so we're not always able to isolate the organism and then get it to cause disease in a healthy individual, either due to methodological limitations or ethical limitations. All right, now, kind of like a slight turn here, we're going to talk briefly about hand hygiene and aseptic technique. The advent of aseptic techniques began sometime in the 1900s. This is when medical professionals started to identify the importance of aseptic technique in medical settings. And just for definition purposes, aseptic essentially means free from contamination. One of the early pioneers here was Joseph Lister. This is Joseph Lister of Listerine fame, or Listerine is named after him. He basically showed that you could sterilize instruments using a compound called carbolic acid, which we usually call phenol, and it helped prevent pus formation from surgeries. And you can see here in this top picture of them spraying the carbolic acid on the surgical field. Then the mother of nursing, Florence Nightingale, was kind of like a exceptional statistician, and she determined that military deaths were primarily due to infectious diseases, right? And so if you look over here, they've got these different colored triangles. This is actually a diagram that Nightingale made, and you've got all the different causes of diseases, or of death, and the blue, I believe, is disease, and then the black is like other causes, and then the red is like battlefield induced like injuries, right? And you can see that there's a much larger area which equates to more individuals dying from disease than they are from either of the other two alone or combined. And because of this, she helped establish aseptic techniques in nursing. I found this kind of an interesting quote, or a useful quote. Every nurse ought to be careful to wash her hands very frequently during the day. If her face too, so much the better. Basically saying that, like, keeping clean is incredibly important for the safety of your patients. Aseptic processes help prevent what are known as healthcare acquired infections, or HAIs. These are also known as nosocomial infections, and they limit the spread of disease, right? And so there are several things that you can do to be aseptic. One is washing your hands. Another would be wearing gloves. Any instruments that are to be used should be sterilized, and all surfaces should be sterilized or should be decontaminated. And so when we are in lab, we're going to practice many of these aseptic techniques. You will wear gloves and wash your hands every day. You will sterilize the instruments that you use, and you will decontaminate your bench surface when you're working. So with that, you should be able to explain the distinction between a pathogen and an opportunistic pathogen. You should be able to compare the theories of biogenesis and spontaneous generation, describe germ theory of disease, and use Koch's postulates of disease. And lastly, you should be able to identify the goals of aseptic technique and explain why it is important in healthcare facilities and laboratories. There is an objectives check quiz available in D2L that you should take to confirm your mastery of these objectives. Thank you. Have a great day.